Hereby I open this academic ceremony at Maastricht University. My name is Ton de Goey. I am the pro-rector of the ceremony and chair of the degree committee. First of all, I would like to welcome Mr. Carlos Peniche Silva. He will defend in public his thesis, which is titled Advancing Tendon-to-Bone and Enthesis Repair from Biomimetic Materials to microRNA Modulation. Welcome all members of the degree committee, not only present here but also present online. And I would like to introduce now the two supervisors to you. The first supervisor is Professor Martijn van Griensven. He is Professor of Regenerative Medicine at Maastricht University. And the second supervisor is Professor Elizabeth Rosada Balmayor, and she is Professor of Experimental Orthopedics and Trauma Surgery at the University Clinic of RWTH Aachen University in Germany. I will introduce the 5 opponents later during the ceremony. Welcome to you here present in the aula, and also I would like to welcome all followers of the live stream. May I invite you, Mr. Peniche Silva, to give a summary of your thesis. The floor is yours. I wish you success in the coming hour.
Thank you, dear Prorector. Thank you, all members of the Corona, family and friends. In the next 15 minutes, I will give a brief overview of my PhD research work, Advancing Tendon-to-Bone and Enthesis Repair. And I would like to start my presentation talking about movement. This is the act of or process of moving, change or place, position or posture. And we tend to give movement for granted. However, within our bodies, movement is the result of the coordinated actuation of a highly specialized musculoskeletal system. The 3 key main players of this musculoskeletal system are muscles, tendons, and bones. And I am most interested in tendons, and in particular, where the tendon meets the bone. And it's that tendons connect muscles and bones in such a way that upon muscle construction, the tendon pulls the bone, which generates joint movement. Tendons as well stabilize joints during movement, preventing joint dislocation. In particular, the connection between tendons and bones, called or termed as enthesis, is a highly interesting tissue. This histology image shows how a soft and elastic tendon connects gradually to a stiff and hard material, which is the bone. In order for this connection to be effective, the enthesis presents opposing gradients of collagen alignment and mineralization that allow for the smooth stress transfer between tendons and bones. Due to the relevance of this tissue in movement, it is of course prone to injuries, and regrettably, during the healing process of the enthesis, it tends to fail to regenerate the native morphology. And the scar tissue that is formed in its place exhibits poor mechanical properties, which makes this tissue prone to re-injuries. So tissue engineering carries the promises of developing new and more sophisticated strategies to improve the healing process of injured enthesis. And for this, normally tissue engineers combine strategies based on the use of biomaterials, cells, and bioactive molecules in order to create bioactive constructs that will allow for the extracellular matrix deposition and a better development of the tissue after injury instead of the scar tissue formation. For this, biomaterials should be biocompatible, biodegradable, biomimetic. Stem cells are a great source of cells for tissue engineering applications, as they are able to differentiate into many different cell types or tissues. And the cherry on top of these approaches is typically the bioactive molecules. These are molecules that can be used to enhance the regenerative capacity of these constructs. So following a tissue engineering approach during my thesis, I first evaluated a plethora of candidates of biomaterials to be used in tissue engineering applications focused on tendons and enthesis, with a specific focus on silk-based biomaterials. Thereafter, I evaluated 3 possible anatomical localizations to explore the tissue engineering approaches that we wanted to develop. And consequently, we selected one of them, created an injury, and we treated it with a scaffold specifically designed to mimic the tendons and the bone interface. In Chapter 5, we look to potential sources of these bioactive molecules. For this, we aim to look at the microRNAs. These are fascinating molecules as they are able to regulate or modulate the gene expression, therefore deciding the fate of cells or even tissues. And finally, we evaluated the possibility to use these microRNAs as potential therapeutic tools in the differentiation of tendons for stem cells. So coming back to our material of choice, silk is known as the queen of fibers. It exhibits extremely good mechanical properties, which makes it suitable for enthesis tissue engineering. Of the two proteins composing the silk, we have fibrin and sericin. Fibrin is the most commonly used in biomaterial application. It is stable, biocompatible, allows for tunable mechanical properties, is permeable to oxygen and water, and is very versatile. So, happy with our selections of the biomaterial, we move on to our animal model. For this, we look into the three most clinically relevant sites of enthesis injuries, which are often the shoulder, the knee, and the heel. We compare this localization in terms of morphology, gene expression, and mechanical properties. And when looking closely to the morphology of these three insertion sites, we identified important differences in the fibrocartilage transition between the tendon and the bone on each one of these anatomical localizations. We additionally observed that the fibers coming from the tendon to the bone are oriented differently in each one of the localizations, which highlights the fact that even those three entheses are indeed the connection point between tendons and bones, they are not created equally. The gene expression profile is represented in these randomly looking graphs. What I want to show here is how different these graphs were from each other. This highlights the differences between the molecular environment of these entheses. When looking at the mechanical properties, it was evident to us that the patellar tendon enthesis was the strongest, closely followed by the Achilles tendon enthesis. And the patellar enthesis showed the biggest cross-sectional area, which is a great thing to have when we work with small animal models, as it allows for better manipulation and easier accessibility when it comes to the creation of defects or implantation of materials. And this is exactly what we did in the next step of my thesis. So we created a defect in an enthesis rat patellar, and we treated it with two versions of a silk fibroin scaffold. This scaffold was carefully designed to mimic tendons and bone sites of the enthesis, with the difference that the SMT label here scaffold showed a smooth and interconnected transition between the tendon and the bone face, while the SAT scaffold showed a more abrupt transition between the faces. When treating the animals with both versions, we show that the presence of this smooth interconnected transition increased the quality of healing of the animals treated with it by increasing the recapitulation of the insertion site from tendon to bone. Additionally, the biomechanic features of the animals were improved, and the expression of important collagen markers and other extracellular markers necessary for the proper healing of the entheses and tendons were upregulated, all while maintaining to a minimum the occurrence of ossification at the tendon side of the entheses. This was not the case when we used an abrupt transition design scaffold. So we then aim to understand better the process of healing, looking a little bit closer to the occurrence of fibrosis. So we came back to our animal model, and we created an injury. In the first slide here, you can see how the native morphology of the enthesis looks like. After the creation of our injury, 10 days after to be precise, the rats develop a significant proportion of fibrotic tissue deposited on top of the tendon, and even the presence of some ossification on the tendon side of the enthesis was visible. So we aim to understand why is this happening, or at least how this can be regulated. For this, we look into microRNAs. As I mentioned before, I'm fascinated by these molecules. They are naturally occurring, which means that we have them in our cells regulating many different pathways. And by regulating these different pathways, they can participate in proliferation, differentiation, or fibrosis occurrence. So we look into the expression profiles of microRNAs that are identified in the literature to be related to fibrotic processes. We look into these microRNAs, and we also look into the target genes that they are predicted to target, or at least reported to be targeted. And when we look in our samples, what we observe is that many of these microRNAs were dysregulated. And in particular, the microRNA STIM5P was upregulated when the reported target for this microRNA in tendon tissue, which was SMAT3, was downregulated. This makes us pay a little bit more attention to this phenomenon and to question ourselves, first, where exactly this microRNA is being upregulated. For this, we performed in situ hybridization studies that allowed for this specific identification of the portion of the tissue that is expressing the microRNA. And as you can see, highlighted here with a blue staining, microRNA was present at the fibrotic portion of the scar tissue. By mathematical prediction models, we identified that indeed SMAD3 was one of the primary targets for this microRNA. And the SMAD3 is very important for tissue engineering, and especially bones and tendons. And this is because SMAD3 intervenes in a very important pathway for tendons. It's the TGF-beta canonical pathway. The activation of this pathway tends to yield to the expression of profibrotic genes, which participate in the scar formation. So our idea then was to utilize the microRNA 16-5P to modulate this pathway, avoiding, hopefully, the scar formation. To evaluate if this was actually possible, we created a tendon-mimetic microenvironment where to test the activity of the microRNAs on stem cells. We loaded these gels with microRNAs, stem cells, and magnetically responsive microfibers. These microfibers will be aligned toward, in a parallel alignment, similar to what we observe in the tendon tissue, under the influence of an external magnetic field. This alignment will give the stem cells encapsulated in the gels the cues to start the process of tendon differentiation. And the presence of the microRNA will either be detrimental or be in favor of this process. That is precisely what we wanted to evaluate. And for this, we not only included microRNAs, but we also included inhibitors of microRNAs so that we could compare. So the cells encapsulated in the gels, they actually managed to uptake the RNA from the gel. This is highlighted here by the presence of these fluorescent green dots inside the cell cytoplasms. When looking at the expression profiles of the SMAD3 in our samples, we also observe that in the presence of the microRNA 16-5P, the expression of SMAD3 was downregulated for at least the first 15 days of culture, and it was upregulated in the presence of the inhibitor. We also look to tendon markers to evaluate the process of tenogenic differentiation of the encapsulated cells. And we observe, indeed, that the expression of tenascin, tenomodulin, decorin, and other important tendon markers were upregulated, whereas in the presence of the inhibitor were downregulated. And finally, the expression of collagen 3 was slightly decreased with the presence of microRNA 16-5P, highlighting the antifibrotic role of this microRNA in this setting. So the main conclusions of my thesis, then, include that, of course, silk remains to be a suitable material for tendon and thesis tissue engineering applications. That in a small rodent, the patellar enthesis is the best localization when it comes to implantations or creations of injuries and so on. That the healing is dependent on scaffold morphology, and that in an interconnected transition between the tendon and the bony phase in this construct is a desirable feature when it comes to treating patellar enthesis defects. And finally, that microRNA 16-5P could potentially aid the process of healing and regeneration of the enthesis by regulating the TGF-beta pathway. With this, I would like to finish my presentation for today. Of course, thanking to the amazing support of my supervisors and to all my colleagues from the Merlin Institute, my colleagues from the 3B Research Institute, and my colleagues from the Mayo Clinic. Thank you all for your attention, and now I will give the word back to the prorector.
Thank you very much for this clear overview of your thesis. The opposition will be opened by the chair of the Thesis Assessment Committee, Professor Roman Druckenmüller. He is a professor of microtissue engineering at Maastricht University. Professor Druckenmüller.
Thank you, Prorector. Dear candidate, please accept my congratulations on your thesis, which I very much enjoyed reading and where I learned a lot. I would also like to extend my congratulations to the supervisor and promoter team. And now let us go to the questions and discussion. My first question concerns Proposition 7, where I would be grateful if one of the esteemed panelists could read this proposition for us. So Proposition 7.
Biomaterial research could benefit from less diversification of biomaterial candidates and deeper exploration of the ones we already have.
Thank you. Dear candidate, can you elaborate in more detail how biomaterials exploration could be more deepened on corresponding methods or procedures to help you in that?
Highly esteemed opponent, thank you very much for your kind words and, of course, for the question. Yeah, when it came to me, the idea of writing this proposition, I was most focused on how evident is in the literature that the quality and the impact of many biomaterials that are already available can be enhanced. And even then, they don't bring enough attention to, at least to my way of seeing it. They don't bring enough attention, and there seems to be kind of a race of see who creates the most sophisticated or the most advanced new material. When it comes, for instance, to the seal fibroin, which is the one that we use here, there are so many ways that we can make this material even better. And in my consideration, it should bring more attention to the biomaterial scientist community.
Thank you. Any ideas how you could ease finding of biomaterial candidates which are already around?
Yes. Well, first, respecting the literature that we have already available, in the sense that it's there to provide the knowledge of how the potential use of these biomaterials. For instance, when it comes with silk, there are surface modifications such as heparin, which our group has already attempted, has shown to increase the retention of growth factors that can improve the regeneration of tissues. Modifying the materials with RGDs peptides, for instance, to improve the attachment site to cells. To make new formulations of the same biomaterials that allow for the production of more elastic materials that mimic better the tendon portion, for instance, when it comes to the creation of biphasic scaffolds and so on.
Thank you. My next question concerns alternative ways of exploration or testing of biomaterials, in particular in conjunction with the biphasic silk fibroin scaffolds, which you applied. As you might know, as an extension of the concept of healthy and diseased tissue models based on microfluidic organs on chips, Merlin PIs were also proposing researching the testing of biomaterials and structures thereof on chip. So if you were to build an on-chip model of a tendon-to-bone enthesis defect, how would you approach it? What essential cell, ECM, and other components would you incorporate, keeping in mind that the complexity of such models is limited for feasibility reasons? So what cells, what ECM would you choose for an enthesis model, or maybe even from the very beginning a defect model, or you would maybe create the defect later on chip? So what would be your choices?
Yeah, well, I will start by first defining which portion of the enthesis indeed we want to deal with. So my cover here represents a Cuban bish, but it's represented in a 4-layer phase, right? Representing the bony phase of the enthesis, the mineralized fibrocartilage phase, the non-mineralized fibrocartilage, and the tendon phase. So when it comes to the design of a bioreactor that will deal with the enthesis in general, we will have to first question ourselves in what we want to focus. Thinking of in a biophasic approach, where we will have then the bone and the tendon, which kind of is an oversimplification of what we have in the enthesis. I will focus on first the collagen matrix. The collagen present on the enthesis will be at the tendon and the bony sides, mostly collagen 1, and on the interface, collagen 2. And this could be actually recreated in vitro models or in even bioreactor models. The cell source to use, I will always advocate for stem cells. However, we know that the cells proliferating in these different phases are typically first in the tendon phase, tenocytes or fibroblasts, then chondrocytes, and finally bones. So we could use, to some extent, fibroblasts as well to improve the tenogenic, the expression of tenogenic markers on the tendon portion, while the HMSCs will be directed to osteogenic processes in the bioreactor.
Mm-hmm. Thank you. I was also interested in a certain mentioning in the general discussion that you mentioned that interface tissues are remarkably difficult to study. Such difficulties arises from many angles, and one of the most overlooked ones has to do with a delimitation of the tissue per se. Considering these difficulties, what does that mean for designing biological robust gradient scaffolds?
Yeah.
So just in terms of, of dimensions and where should be what? Yeah,
that's a very good question. And what I meant there was that in contrast with tendons or bones, which are easily identifiable, the enthesis is a gradient. So when it comes to explore the actual nature of the enthesis, we should set, or it would be a good idea to set limits to where the enthesis starts or begins. And this is, of course, will have to be correlated to the design of the material that we want to use or the construct that we will use on treating defects on the enthesis. So it was more about the standardization methods that we will have to come with to delimit a certain distance from, for example, the fibrocartilage transition, so that we know that at least until this point is still enthesis and then becomes bone, or until this point is enthesis and then becomes bone.
Dear candidate, thank you. Dear Prorector, I'm very much satisfied with the answers of the candidate, have no further questions, and would like to give back to you the opposition. Thank you.
Thank you, Professor Trockenmüller. The next opponent is Professor Denise Ducheva. She's Professor of Regeneration of Musculoskeletal Tissues at the Department of Musculoskeletal Tissue Regeneration at the University of Würzburg in Germany. Professor Ducheva.
Thank you, Prorector, for this kind introduction. Dear candidate, I first would like to express that I read your thesis with great interest, and I particularly appreciated the very logic build-up and the very high quality of the scientific exposé. Compliments to this work. My first question will be based on the proposition number 1, and here I kindly ask one of the paronyms to declare it.
To understand the injury tissue, we must first understand the healthy one.
Thank you. In your chapter 3, you characterize entheses at different anatomical locations. I was very interested in the result showing that the patella and the Achilles have similar cross-sectional area, however, a different cross-sectional area, however, similar biomechanical behavior. So how do you explain this? What are the determinants of this behavior?
Highly esteemed opponent, thank you very much for your question and for your presence today in my defense. This is a very interesting question, and of course we ask this same question ourselves when we look at the data. Indeed, the patella showed twice as bigger size of the cross-sectional area as the Achilles tendon. However, when it comes to mechanics, it's not only the area what matters here, but also the transmission of force from the tendon to the bone part. And in the case of the Achilles showed the longest associated tendon, which allows for a more effective dissipation of energy when it comes to pulling. Another possible explanation will be that the interface that we observe in the Achilles tendon was a slightly more abrupt interface between the tendons and bone, and some other mechanical studies have reported that such a type of integration into of the tendon into the bone is more effective when it comes to the transmission of force. And I will say that perhaps the third point that could be also playing a part here was the high expression levels of collagens that we found in the Achilles tendon compared to the patella. If you look closely, the collagen 1 which is the main component of the enthesis and is the most the main response. It bears the responsibility of conferring the mechanical strength to the tissue. Was in the Achilles tendon was significantly higher than in both the patellar and the supraspinatus tendon.
Very good. Have you looked also at elastin content?
No, we didn't do it.
But do you think it matters?
Yeah, it could definitely matter. Of course, it will be closely related to the ability of the tendon, especially to dissipate energy.
I would like to proceed to my second question, which will be proposition number 4. And here, thank you.
Heterotopic ossification at the tendon-to-bone enthesis is, at least for now, inevitable.
It is very interesting that tendon injury, very often cartilage lesions, and so enthesis, when experience injury, responds with heterotopic ossification during the repair process. So what are the cellular bases of this process, and why do you think it's kind of the default mechanism in the tissues to occur?
Yeah, this is also a very interesting question, of course. So I will start defining that at least what we have observed and most of the evidence reported in the literature of ossification happening in tendon is that it occurs chondrogenic, so via the cartilage mineralization on the tendon. So it's a chondrogenic ossification. And the chondrogenic ossification has to do mostly with, or is very evident in overload, so in sites that suffer from a lot of overload, which increase the expression of first chondrogenic markers. And thereafter, with time, it will start depositing minerals forming the bone in this area. Now, the problem with the enthesis is that, depending on the of course, the localization, it may be very close to the bone. And the infiltration to the site of the injury from bone cells that can come from either the bone marrow or from the bone itself could elicit the response to start mineralization processes. I was also thinking on the surgical approaches that are usually practiced when it comes to the surgical handling of these injuries. They often involve sutures, and these sutures through the tendon have shown to increase the occurrence of ossification or mineralization at the site of the suture. So even though these portions of the tendons may not have been injured initially, the way that we have, or at least the surgical strategies that we have are available, they may be effective to connect the tendons and bones, but they will create, with time, stress concentration points due to the sutures that will eventually create ossification.
The altered biomechanics might be one of the driver.
Sorry?
The altered biomechanical environment.
Exactly.
But now, what do you think are the cell types kind of involved in this process? Who can be the source of this heterotopic ossification?
It could be, and this is a speculation of mine because we didn't look into specific cell types, but at the cell level, we see a lot of infiltration of inflammatory cells. And it could be that the process of macrophages infiltration could eventually lead to ossification or osteogenic differentiation of the macrophages, and even potentially the tendon progenitor stem cells, that they could also become bone cells.
Very good. I think I'm very satisfied actually with the questions, and therefore I will now further proceed, and I can give it back to the prorector. Thank you.
You have more time, but if so, we go to the next opponent, and that is Professor Dimitrios Zevgolis. He's not here, but he's present online. And he's professor of regenerative medicine in the School of Mechanical and Materials Engineering at the University College Dublin in Ireland. Professor Zevgolis, the floor is yours.
Dear Prorector, thank you very much. Dear candidate, dear committee, thank you for the invitation. I'll start with congratulating the candidate for this wonderful piece of work, and obviously the supervising team from bringing you to that level of becoming a young and independent scientist. I have some questions, some technical questions and a philosophical question in the end. I'll start with a question that came to me through the presentation. Silk, you mentioned, is the queen of the fibers. Dare I ask who is the king?
That is a good one. Highly esteemed opponent, thank you very much for your kind words on the question. Well, it could be collagen.
Thank you. It was just curiosity. Then I'll go to a technical question. There are certain gene analysis data in the thesis. For some genes, the values were below twofold. What is your opinion on the validity of fold values below 2? How accurate are they? How sensitive is the experiment? And therefore, how able are we to draw safe conclusions for fold values below 2?
Thank you very much for the question. This indeed was also a concern of us when we were working with the gene expression profiles, and therefore we decided to set cutoff values of 2-fold, and just to consider or to use it to filter results. I think that the techniques that we have still ongoing to validate or to evaluate the expression of genes, even though they are extremely reliable, the calculation methods and the errors coming from not only pipetting errors when it comes to the selection of a certain, of the mounting a plate for PCRs, and just to make clear that we are talking here about the PCRs that we conducted. The errors from the operator, but also the errors coming from the machine, and even some errors that may come from the use of perhaps not the ideal housekeeper gene when it comes to afterwards the validation and the calculation of the fold change could indeed account for small variations of the fold change. And that is why, in order to avoid such errors, to have them in consideration, we selected higher CT or fold change cutoff, just to avoid that we will be misinterpreting the results. I don't know if this exactly.
Thank you. The second question is again experimentally related. I believe on the chapter number 4 on the preclinical data, through the microscopy techniques following the picrocereus red staining, you have evidence some collagen type 3. How do you think that could have possibly affected the results, the healing capacity of the tissue?
Thank you very much also for pointing out this finding. We observed indeed a disorganized matrix of collagen in the site of the injury there. And of course, the presence then of collagen 3 was evident in the site of the injury. And collagen 3 is typically a short, so compared especially to collagen 1, is a short fibers that interconnect in between collagen 1 fibers, and is very poor in terms of mechanical strength. Therefore, a way of assessing how well, let's say, a tendon or enthesis is being regenerated in injury, we can consider the ratios between collagen 3 and 1 as an indication of a better or a worse regeneration of the tissue. The highest presence of collagen 3 is usually then correlated to low mechanical properties, and this is mainly due to the lack of alignment when it comes to the native structure of the enthesis or the tendon.
With respect to ossification, could that be the naive nature of the implanted cells? So MSCs, they have the inherent capacity to differentiate towards osteogenic lesions, among others. Could ossification be attributed to an unwanted differentiation of the MSCs?
It could be. We didn't add HMSCs in our constructs, so we don't have evidence that making responsibles of the HMSCs for this ossification when happened. But definitely, we have seen that even during cell culture, HMSCs tend to undergo or tend to go easier to ossification than to other differentiate to other tissues. So yes, indeed, they could play an important role on ossification.
My last technical question is the use of the two different scaffolds. So the first chapters are focused around the silk, while the last one is the GelMAC, the hydrogel, if I'm not mistaken. Why did you choose to change the scaffold? And if you have to do it all over again, will you have used a different scaffold composition or structural wise?
Much also for that question. So I wouldn't change it. So I think that I was very satisfied with the way that we did it, and it's especially because the point of using GELMA was not to create indeed a scaffold, as the scaffold of silk fibroin was. It was more to create a model that we could easily control in terms of imaging, in terms of encapsulating materials that the cells will have then to their disposition to uptake. It was also for the availability of these microfibers that were magnetically responsive, and therefore were giving us the freedom to align these fibers the way we wanted in this case towards tendon. But as you perhaps noticed when reading this chapter, it shows preliminary results. This is it is part of a much bigger idea, where we also want to use the same platform to magnetically stimulate fibers, or to study another type of parameters that studying them in the alternative, which was the silk fibroin scaffold, would have been a little bit more cumbersome and perhaps even less precise.
Thank you. And my last question is more on the philosophical side of the tissue engineering regenerative medicine. Tendon injuries, they are not life-threatening conditions. We have seen the last few years during the pandemic, people were very resilient in mRNA delivery and the vaccination. So what is your opinion on using gene therapy in tendon and in general in non-life-threatening conditions?
So this is also a very interesting question, and I'm happy that you bring it up, because even though I didn't use gene therapy per se in my tissue engineering approach, I have collaborated with, and I'm very proud to have collaborated with many with other gene therapy approaches, especially focused on bone. And I think that the stigma surrounding gene therapy is slightly being overcome by the gain of knowledge about the therapy and the gain of knowledge of the platforms to deliver gene therapy. There was never nothing wrong about gene therapy per se. It was more about the way of implementing the therapy, specifically using viruses as vectors of genes. But because, thanks or not to COVID pandemic, this meant not only a huge improvement in the technology, but also improved the awareness of the community about the potential uses of the gene therapy, as we all have now a little bit of gene therapy on us. So I expect that with the gain in knowledge about the technology and the gain experience, as this, I think that it will be the biggest human trial ever carried out, if we count the vaccines for COVID, I think that this will definitely catapult gene therapy, not only in tendons and bones, but to many other scenarios.
Thank you. Thank you very much. Again, back to the pro-rector, I suppose. I'm satisfied with the answers, and I don't have any more questions.
Thank you very much, Professor Zevgulis. There would be time for another question, but if not, we move on to the next opponent, Dr. Berta Chilero-Pastor. She is Associate Professor for Spatial Omics for Regenerative Medicine at Maastricht University. Professor Chilero-Pastor.
Thank you very much, Prorector. Dear candidate, I would like to congratulate you and your supervisors for this very nice thesis. I find fascinating that you really did a lot of fundamental research, but then more and more through the thesis, you really move into clinical applications and into therapy. So congratulations for that. So my very first question is on chapter 2, because you do this in-depth literature review about different biomaterials for tendon repair. And I found interesting that I think that you forgot a very important element in this, and these are the cells, right? So you did not describe what could be the role of cells, no? The cell biomaterial interaction, depending on the location, for instance, of the injury. So can you elaborate on that, and why did you decide to basically not describe the role of the cells in this tendon repair?
Esteemed opponent, thank you very much for your question. And yes, indeed. So first, the scope of the chapter and the scope of the review, of course, was selecting the biomaterial. And especially the biomaterial of choice here, the silk fibroin, was based not only or not because of cells interactions in particular, but because of the availability, the easily handling of this material, the versatility and the biocompatibility and so on. But even though we didn't have, or we don't have it in the book, we have a lot of experience working with cells on these biomaterials, and specifically with silk fibroin. We evaluated that the ability of cells, and when I mention cells here, is talking mostly about stem cells, especially adipose-derived stem cells. They seem to really grow very well on these materials, and to even read the morphological cues provided by the biomaterial design. So before before deciding to use this silk biomaterial in our animal model, we did do, and this was work from one of the previous PhDs of Martijn, we did do a lot of evaluations of cell proliferation and differentiation on these materials. And it was not, even without modifications like RGD modifications on the surface and so on, cells seem to really integrate in the material and to differentiate towards tendons and bones, regardless the extra modifications. We also evaluated, as I mentioned, with our previous discussion with Professor Roman, modifications of heparin. These modifications of heparin to deliver growth factors was indeed promoted even better integration when it comes to, not integration, but better differentiation of the cells when providing the growth factors to differentiate into chondrogenesis or tenogenic pathways.
But you focus pretty much on the differentiation aspect.
Yeah.
What about the host response, like the surrounding cells, tenocytes, or infiltration of immune cells? Yeah. So what's your vision on that?
Regrettably, we didn't look into that. So I will say that there is extensive evidence for silk fibroin to be very good in terms of compatibility. And especially fibroin, in contrast with sericin, the other protein that composes silk, is not immunogenic, so doesn't tend to elicit immunogenic responses. So it was not a concern. When we performed the in vivo study that you see with the implantation of the silk fibroin scaffold, one of the things that we didn't see was any signs of beyond normal inflammation or immunogenic response, for at least for the part of the animals. So that's why perhaps we didn't focus or we didn't strengthen that point in the book.
Okay, thank you very much. Now I would like to move to chapter number 3, where you compare the properties of the tendons at different locations, and you use different methods, so molecular techniques, mechanical, the mechanical properties of the different tendons, histology, and if we look at page number 42, you mentioned that for the microscopy measurements, you align the images using the bony parts that you were able to visualize on the samples. So my question is, have you used as one single point for this image co-registration, or how precise is this alignment, right? Because you are looking to the angle of the fibers.
No.
How precise is this? I can imagine that just one single point, you might have some errors, but yeah.
Indeed, yeah, one single point will give some errors. That is why we actually defined 3 different regions of interest to measure the alignment. So what we did is that we divided these regions of interest covering the whole width of the tendon, And using plugins from Imagery, we calculated the alignment of the fibers in each one of the three regions of interest. And those are the values that are reported. So to finish this idea, then, the alignment that I am reporting there is the average that we found in each one of the three regions. But it was very interesting to us that in the case of the supraspinatus, the differences of alignment in these three different regions that we define was actually quite significant compared to the differences in the patellar and the Achilles tendon. This highlights that because of the shape of them, you could think of the patellar and the Achilles tendon as more rectangular in shape, where the fibers insert kind of homogeneous into the tendon. But when we look at the supraspinatus, as is in a more round scenario, and the tendon moves, let's say that it covers a more round surface, then the variation of angles was widest here. So we saw a very significant variation of the angulation, and most likely because of the shape of the bone or where the tendon is inserted. So that is why we consider the entire width of the tendon. Otherwise, one single point would have brought
I understand, but let's say if you use just one single point, you are saying that you are using the average, right? But if the alignment is not totally correct or the co-restraining is not totally correct, the average will have also a defect, right? So it will not be very precise. So I was just wondering whether it was not possible to technically include maybe something like fiducial markers. You can use maybe to inject ink at different locations of the block, because I assume you have used FFP blocks, I'm not sure, with paraffin. And then you could use maybe 3 different points for the alignment. Or this was not possible from a technical point of view? Or?
I think I understand your point here. And in our case, it wasn't necessary because we did the calculations on the picroserious staining images. And what was good about this is that with the picroserious, and these are healthy samples, so we can have a very clear visualization of the fibers. So what we did is that in the 3 entheses, I selected as an horizontal line the interface with the bone. And we positioned the samples to match the horizontal line at the insertion point. And then the entire region of interest that we selected was not a point, but was actually including many different fibers. So the region will compute the alignment of a group of fibers, all of which were enclosed by the region of interest that we selected. So it was not even a single fiber, it was a group of fibers. And therefore you will have the average when it comes to the 3 regions. Now, of course, since the differences that I was mentioning before between the supraspinatus and the other ones was so high, the average then of the supraspinatus tendon enthesis showed a biggest variability when it comes to the standard deviation. There you will see that localization in particular showed a quite significant variability when it comes to the insertion angles. Okay,
thank you for your answer. Now I would like to move to page number 43, where you describe that for gene expression analysis you have done a bulk RNA extraction. And then if we look into figure number 6, you do see some variability in gene expression in the different samples, in different animals. So I was wondering whether this is due to the differences between animals, or you think that because you are using a bulk RNA expression technique, of course, maybe, where you do this RNA extraction, how much you take from one area or the other, it really affects the measurement. So can you please elaborate on that?
Yep. Yes, of course. So first, I think that the variability may come from the selection of the isolation of the enthesis samples per se. So as you see in histology, you see the entire unit of tendon, fibrocartilage, and bone. But when it comes to the genes, because we wanted to focalize in the enthesis, we had to actually manually trim down the tendons and the bone. So it was very hard to standardize how much, and this was also part of the discussion that we had with Professor Roman before, so how much of the enthesis, the tendon or the bone we were taking. Since the enthesis presents this gradient of cell populations that may be expressed in different markers, then this could eventually affect the PCR results when you have specific samples that contribute a little bit more with the tendon portion of the tissue and another ones with a little bit more of bone. So I think that they are more related to this phenomenon.
Okay. So overall the animals were, behaving in a similar way.
Yeah, they were all healthy animals, and the treatment. So these animals, indeed, they were not treated for anything related to tendons or musculoskeletal defects. So I don't think that variability between the animals could have affected greatly here.
So perhaps here methods like laser microdissection to really cut exactly what you really want to look at, and then RNA extraction might be helpful to kind of validate your data, perhaps.
Yeah,
that would be
indeed.
an idea. I think I have time for one more question. Yeah. So then, I think I'm gonna move to chapter 6 then. You have used here adipose-derived stem cells. And you have very nice results, but I would like to know whether you could also consider the use of other adipose-like stem cells that might perhaps behave totally differently. For instance, phosphat as a source of adipose cells, and stem cells in this case. Could you please perhaps reflect on the importance of the source of stem cells to really be used as a therapy?
Yep.
Or do you think that this would not play any role and you can take them from wherever?
Just to make sure that I understood the question, we are talking now about the sources for the stem cells.
Exactly.
Okay. Yeah, no, indeed, I think that they actually play a very important role. Even though they are typically considered to be pluripotent, all of them, they can be more resilient to undergo a certain differentiation or another one. And we have seen that in the labs. Especially when it comes, and this is an example that I can give you from experience myself. I have worked as well with the bone marrow derived stem cells. And when it comes to the transfections and the gene modulations with microRNAs, which is part of what I'm very interested in to do, we have seen that in adipose-derived stem cells, the regulations of the genes that we have attempted to regulate occur more effectively and at earlier time points than with the bone marrow derived stem cells. This could be one of many other examples of how, depending the cell source, they could be more prone to uptake or to differentiate towards a certain pathway. In this case, adipose-derived stem cells was a very good source for us, since we were getting cells directly from the clinic, of course, with the approval required. And they are extremely easy to isolate as well, and they grow very fast. And we have a lot of experience with them, therefore we use them.
Okay, thank you very much. I'm very pleased with your answers, and I give back the word to the prorector.
Thank you very much, Dr. Berta Silero Pastor, and thank you for being the secretary of this committee. The fifth and last opponent is Dr. Pieter Eemans. He is Associate Professor for Orthopedics at Maastricht University Medical Center Plus. Professor Eemans, floor is yours.
Dear candidate, also my compliments to the work and your team. I think a lot of techniques are exploited there, from basic fundamental research to histology to surgery. And allow me to ask some questions from the clinic downwards to the fundamental part. So first of all, did you do the surgery yourself?
Esteemed opponent, thank you very much for your words and your question. Unfortunately, no, I didn't perform the surgeries.
Okay, and did you have a look at the animals after surgery? What happens to a surgical field? What happens to the environment? Or did you ever have a scar yourself? And what happens to such an environment?
Yeah, well, in such environment, so the first part of the question.
Yeah.
So I couldn't follow closely the animals, as this was done by one of our collaborators in the Mayo Clinic, the surgical part. But yeah, I'm aware of how the body tends to respond, at least a healthy body. And it starts with inflammation and, of course, the deposition of fibrotic tissue. Eventually, if we are lucky enough, with the years, this fibrotic tissue will become, will remodel towards the native or more native-looking tissue.
So that's a great start for the rest of my questions. So I'd like to take you then to page 71, where we see the example of the heterotopic ossification. And can you tell me a little bit after you do surgery, because you already started a little bit with my colleague, sitting in front of me, that you answered the sutures might give an effect. Can you tell me something about the relation of inflammation and heterotopic ossification? Did you dive into that? Do you know anything about the relation between, well, surgery provokes inflammation, but do you see differences there? Do you think that there's a correlation between inflammation and heterotopic ossification?
Yep, thank you very much for the question. So, as you see in the animals with on the presence of ossification, one of the highest that was not treated by the scaffold was the TC group, transversal cut group. Mm-hmm. This group showed a lot of ossification, mm-hmm. and at the same time, this group was suffered a lot from sutures, mm-hmm. because the way this group was treated was that a defect was created at the tendon side of the enthesis. So the enthesis part was preserved. It was only at the tendon side. Mm-hmm. And the only way of connecting them together, the tendon side, was by performing sutures. Yep. So therefore, we think that the pressure points created by the presence of the suture here will have triggered the eventual ossification at the tendon portion. Yep. And then when it comes to the possible mechanisms that will be behind the occurrence of ossification in this area, would be definitely related to inflammation. And as I was hypothesizing before, it will most definitely be related to macrophages infiltration and polarization that they could lead eventually to ossification.
No, I'm very pleased with your answer, so I totally agree. But then I would like to ask one of your paronyms to read indeed, it was already with proposition 4. So can one read proposition 4?
Heterotopic ossification at the tendon-to-bone enthesis is, at least for now, inevitable.
Okay, so when doing surgery, we encounter that problem quite a bit, heterotopic ossification, especially with total hip surgery. And we found something that it was not inevitable. And we gave anti-inflammatory drugs like ibuprofen and those kind of things. So we noticed a significant drop, and now with standard that we provide in 2 weeks with NSAIDs to prevent heterotopic ossification. Can you elaborate a little bit on the correlation maybe then, how we can use some of your stuff to mimic those drugs or to implement it maybe in your scaffold to also control some inflammation, because now maybe your proposition 4 may become inevitable. Yeah. So please elaborate how we can do that. Should we provide medication? Can we put that in the scaffold? So I think there are ways to do it, but maybe we can exchange some thoughts. Yeah.
Thank you very much. I unfortunately don't agree entirely with you, even you are in the clinic and I am in the lab.
Very good. Very good.
And I think that my position here is because I'm trying to see the patient in the lifetime. Mm-hmm. And even though we could minimize the occurrence of the punctual occurrence of ossification after a certain treatment, as it may be, for instance, the hip replacement that you mentioned, or via the presence of anti-inflammatory drugs after a tendon treatment or enthesis treatment, I do think that ossification in the long term may appear as the result of the natural process of degeneration of the tendons or enthesis. And because of microfractures that may occur that we, even during the practice of sports and so on, that we may not even be aware of, but will slightly become to, so it will slightly begin to form ossification at the points of these microfractures. Of course, I will acknowledge that the approach that you are using now is a very clever one, and I will dare to imitate it with the microRNAs that we were profiling. So the microRNAs that we profile at the enthesis side of the injury, some of them were strongly antifibrotic but anti-inflammatory as well. And this is the case, for instance, of the microRNA 16-5P. So in my presentation, I mentioned that it targets SMAD3, but it has been related also to targeting interleukin 1B and interleukin 6 as well, which could then act as an anti-inflammatory microRNA that could prevent the overexpression of cytokines that could drive inflammation and therefore improper healing of the enthesis.
No, that's an interesting solution. So my last question is, if you look at old age, because your first proposition goes about the look at the normal, healthy tissue, but of course there's a reason why a tendon ruptures, and often that's the case, which you mentioned, small cracks, older age. So how are older cracks during older age differentiating my knife? So that's also a big crack. Both give inflammation, right?
Yeah, indeed.
And would it not be good as a follow-up then to study this in a more inflammatory environment, which might cause the heterotopic ossification as well as the degeneration of the tendon, maybe caused by overuse, because it's a chronic overuse and I do it in one hit. So we do it in one hit, but nature does it slowly.
Yeah.
So can you elaborate a little bit on that, how you would look also to an inflammatory environment? And then it's the opposite, because then you don't want to give maybe too much anti-inflammatory drugs or, yeah.
Yeah, indeed. Yeah, that's a very interesting point. And in my case, I think that I would like to continue looking at the microRNAs as.
Yes, Mr. Peniche Silva, the time for defending your thesis has passed. The committee will now withdraw to discuss the quality of your thesis and in particular the quality of your defense, and please await our return.
All right, please.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
My goals only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. I know what's on the line. I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Take the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. Southside, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goals only getting clearer, east side to the west side. No place like home. If it's questions that you've got, go the extra mile and die. can't fly by now, don't waste all your time. Cause I'll go the extra mile.
Mr. Peniche Silva. The degree committee here present, and also not only here but also online, has assessed the quality of your thesis and in particular the quality of your defense. And in view of its positive verdict and taking into account your previous qualifications, the committee has decided to grant you the degree of doctor. And Professor van Griensven is authorized to confer upon you this academic distinction in accordance with Dutch university custom and law. I invite now your supervisor now to take the floor. Professor van Griensven.
Do you promise to work in accordance with the principles of scientific integrity at all times? To be careful and honest, transparent, independent, and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Carlos Julio Peniche Silva, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university. Dear Dr. Peniche Silva, what an honor to be the first one to address you like this. I am extremely proud today. You are my first student that completed 4 years working in Maastricht and finished brilliantly this PhD. Elizabeth will tell afterwards how you ended up here. In these 4 years, you have made an amazing development. You have become a critically thinking scientist with groundbreaking out-of-the-box ideas. You covered biomaterials, biomechanics, animal experiments, molecular biology, etc. You traveled to Portugal in your car filled with everything needed for the experiments. Of course, missing your wife Brenda, but you said it was worth it, and we saw that today. It is part of your thesis. The writing, however, is a funny part with you. I am very picky with recognizing double spaces, wrong spaces before commas, missing letters, letters that you changed. And you were sometimes thinking faster than typing, I think, and then you were ashamed of the typos. And then you told me, lo siento mucho, profe. You are not only a marvelous scientist, but also a fantastic person. Everybody in Merlin knows you. Was it only because you photographed each of them, right? And you will see afterwards how many people contributed when your paranyms put out an email for your defense. But it's not only science. You also do a lot of sports. From bouldering, road biking, mountain biking, and of course, being a typical Cuban, skiing. Speed is really what you like. And you have little accidents with hematomas and small deformations on your fingers that you show me then, but you keep on going. By the way, biking. You have many bikes and are actually a very good bike mechanic. When you refurbished, together with Ralt, the 40-year-old bike of my dad, the race bike, this was a great surprise, both for my father and for me. And we'd like to thank you for that. And this year, I am sure it will participate in the Aeroica. When I was writing this laudatio, I realized you are really integrated well in the Netherlands. Not only you are an avid fietser, but you and Brenda also bought a rijtjeshuis, where you live with Brenda and Leito, who is as crazy as you are sometimes. También quiero felicitar a tu esposa Brenda, que está siempre a tu lado, apoyándote. Las palabras en tu tesis dedica a ella muestran el intenso amor que ustedes se tienen el uno al otro. A tus padres, Marlene y Carlos, que pudieron llegar en tiempo a los Países Bajos, ustedes pueden estar muy orgullosos de su hijo. A tus dos hermanas, Irma y Hazel, y a tu Mimi en Estados Unidos, felicitaciones. Today is a very important day in your professional life. You deserve it. You should enjoy it fully. And I'm very happy that you stick around as postdoc and will enrich our group with further ideas and that you will bring regenerative medicine forward. I am very grateful to know you as a fellow scientist and as a friend. Muchísimas gracias por todo. Solamente me queda expresar mis mejores deseos para ti.
Today, we are celebrating the culmination of a journey full of learning, willingness and dedication, effort. A journey also touched by trust and friendship. Today is your day, Dr. Peniche Silva. And what an amazing journey has brought us here today. We have known each other for many years, as your father was my master thesis supervisor at the time. So you rapidly got my attention when you contacted me asking me for a PhD possibility in our lab at the time in Munich. I didn't have any vacancies at the time, but you worked hard to obtain a DAD scholarship to join our group. Despite a very hostile environment where you experienced bullying and other unprofessional behaviors, you never complained. You always worked very hard, always learned, always help others, although they didn't help you back. After completing 1 year of your PhD in Munich, Martijn and I decided to move to the Netherlands. Martijn was joining a wonderful institute, the Merlin Institute, as a full professor and as a department chair. My only regret was leaving you back. It didn't feel fair. Martijn then decided to offer you the only PhD vacancy that was given to him as a full professor. You didn't even blink. Your answer was instantaneously, yes, you said. I don't care to start all over from the beginning my doctoral thesis as long as I am part of your group. I would like to thank Martijn for the unique opportunity that he has given me to be part of your supervisory team. You are one of the kindest and nicest person I know. And one remarkable, brilliant young scientist who will make us proud for years to come. I would like to thank you, Carlos, for allowing me to get to know you better and to work with you. In your acknowledgement, you say that having us both, Martijn and I, as supervisors was definitely the highest highlight of your PhD. And I would like to give that back to you. Having you as a PhD student is definitely one of the highest highlights of my academic career as a scientist and as an educator. A tus padres y hermanas, y naturalmente a Mimi, quisiera extender mis más sinceras felicitaciones. A ellos van mis deseos de que disfruten de este día y se sientan muy orgullosos de ti. Muchas gracias por la educación que te han dado, por forjar la bellísima persona que tú eres. A tu esposa Brandy, mil gracias por seguir a Carly hasta el fin del mundo y por el apoyo incondicional que siempre le has dado a él, a mí y a Martijn. In closing, I would like to thank all colleagues, family, friends, here and online, for being present in this Defend Act and supporting such a special day in your life. I would like to extend my most sincere gratitude to the members of this committee for taking the time to read this thesis, preparing questions, and being here for you. Carly, today a journey ends, but a new one begins. There is no doubt in my mind that we are standing here today in front of one future leading investigator, one that will conquer any dream and any challenge ahead. Our journey continues. We will continue being part of each other's life, personally and professionally. Congratulations.
Esteemed Dr. Peniche Silva, dear Carlos, it's a great pleasure to congratulate you with your doctorate, and I congratulate you also on behalf of Maastricht University. And I would like to share some impressions we had from the committee. You had a very clear presentation, and as far as your thesis is concerned, we have seen that it's very good in construction of the studies, because these studies form a nice combination of theoretical concepts, techniques, and methods. And I think that that interesting topic that you have studied was quite poorly understood, and I think you have contributed to a better knowledge in this field. And I also think, and the committee thinks, that there are a number of performing your fundamental studies have provided a basis for further studies. And in particular, and it also has been touched by some members of the committee, that it makes then possible to have a much better sight on the combination of going from the bench to bedside. And I think that's important. We appreciated the discussion with you. The discussion also on complex issues was interesting, was honest and clear. And you also very nicely demonstrated that you are very knowledgeable in the field and aspects of your scientific studies that you have performed. So congratulations with that result. And I also would like to congratulate your two supervisors, Professor Martijn van Griensven and Professor Elizabeth Rosado Balmayo. Congratulations with the result in terms of not only the thesis and the defense, but also a list of good publications. I would like to include in my congratulations also your family, your parents, your partner, and other members of the family. And I hope that my Spanish is not so good, but I hope that you understand my English and that you are maybe proud on the result that you have seen here today. Congratulations also to the people here in the audience here in the aula. I think that a number of colleagues and friends are gathered here, and I think that also people following the live stream belong to people that have seen you working very hard in the last couple of years. I would like to thank all the members of the two committees, the Thesis Assessment Committee and also the members of this degree committee. Thank you, and already one of your supervisors said it already, thank you for critically reading the manuscript and having interesting questions and a good scientific discussion. Thank you for that. And before I close this academic session, I would like to ask Professor Dimitrios Zevgolis from Dublin to congratulate you. Dimitrios. Please unmute your microphone.
Can you hear me now?
Yes, we can understand you clearly.
Perfect. Dear Dr. Carlos, what an achievement. Genuinely, what an exceptional PhD examination. The love of your supervisors towards you. Well done, well done. I'm looking forward to working with you and your team, because that is a certainty in the future. Congratulations, Doctor.
Thank you very much.
Thank you, Professor Zevkolis. Before I close this academic ceremony, I would like to ask the close family to stay here with us, and I would like, if we have left this aula, I would like to ask the audience here in the aula to go to the rafter where is the reception. It's quite simple. If you go to the left and you turn to the right after the elevator, you will find the rafter very easily. Here, we will make a picture here with the committee with Professor Zevkolis still on the screen, and then we will go, the committee and the close family, to the hall where we will take another picture on the stairs. Hereby I close this academic ceremony. Don't drop it, then the seal breaks.
Keep the seal well in your hand.
And if then